Hello lovelies, in this video the brilliant Dr. Edwards is going to be taking you through our AQA AS Biology paper 2. Now what she's going to do in this video is show you the questions, show you how to interpret the questions, any clues the examiners are leaving you, so then you can have a go at the question yourself before playing the video again and working through the answers with Dr. Edwards.
Hi everyone! In this video we're going to go through the AQA AS Level Biology Paper 2. So hopefully you've already done Paper 1, had a go at it and marked it and maybe watched that video walkthrough already. So this is for Paper 2. Again, make sure you've sat down, had a go at this paper in exam conditions, and we're going to go through it together and hopefully work out maybe which questions you tripped up on, figure out what the questions were asking us, what content we need to answer this question so that you can identify any knowledge gaps. I'm going to go through the answers and explain the mark scheme in case they didn't make any sense to you, but also point out things you can and can't get marks for so that it can help you mark your work more accurately. Off we go. Okay, so question 1, part A. Explain what is meant by the following terms. So explain what is meant by when it's used as a term to sort of talk about a term is basically the same as saying, can you define these terms? So it's no different from a definition. It's just a fancy way of saying that, and it normally means that there's more than just like a word or a few words. So with both of these are 2 marks, so we need to say enough to get both marks. So it'll be a statement or a phrase that you have learned, hopefully. So a species, this should be easy, we've been doing this since GCSE, is a group of similar organisms that can breed to produce fertile offspring. There's 2 points here. The idea that the group is made up of similar organisms and that they can breed to produce fertile offspring. That's the bit that you need. So a hierarchy for part 2. This is one of those terms that we use to talk about with classification. So this idea of the taxonomic groups that we have form a hierarchy. So it's used to group the groups or to organise the groups that we're putting organisms in to classify them. So the way a hierarchy works is you have groups within groups or large groups divided into smaller groups, and there can be no overlap between those groups. So you can't be in more than one. So you move down the hierarchy. So we know with classification, you can be an animal or you can be a plant. You can't be in either of those. And then underneath that, you can be a vertebrate or an invertebrate. You can't be in both groups. So groups within groups and no overlap between the groups. Giant pandas and red pandas are both mammals found in China. Red pandas were once thought to be close evolutionary relatives of the giant panda because of their shared habitats, bamboo diets, and the similar morphology of the bones in their hands. Using genetic analysis techniques, the following phylogenetic tree was produced. Okay, so we have like a phylogenetic tree, which is a tree that is representative of evolutionary relationships. So it's a way of a taxonomic tree, but it's to do with or has been built around genetic analysis techniques or evidence that talks about DNA. These clearly questions will be focusing on this. If we go to the bottom of the tree, that's obviously the shared common ancestor between all these species. And then you can see the 2 families labelled above that. So the giant panda is there and the red panda is there. They are not on a shared kind of branch. They're not directly next to each other. Using evidence from the diagram, state the closest relative of the polar bear. Okay, so simple question first off, just to see if we understand how to read one of these trees. So we've got the polar bear there, and that is on the closest branch next to it is the brown bear. So clearly the brown bear is the closest relative because they're sharing that branch. They have that shared common ancestor there before they split off. Whereas obviously if they were further apart, then they would be less related, but they are the closest 2 together that share the same branch, so that is the answer for C. For D, does the phylogenetic tree support the earlier conclusion that the giant panda and the red panda are closely related? Explain your answer. So it wants us to look for evidence in the diagram to support this idea that the red panda and the giant panda are close relatives or close evolutionary relatives. So we need to give a yes or a no, and then we need to give reasons why using evidence from this diagram. So we've already said that the giant panda and the red panda are not on the same branch. So the red panda is sharing a branch with the raccoon and they are in the same family. And then the giant panda is in the same family as these other bears. So because they're not sharing a branch, They're not super close evolutionary relatives. So the red panda is more closely related to the raccoon. We've also got this evidence down here, obviously, that they're in different families. So we can say that they're in different families, but what we can say is that they are related evolutionarily because they do share this common ancestor. But we can't say they're close or very close evolutionary relatives because they're in different families and because we've got evidence that the red panda is closely related to something like the raccoon, which is in the same family and they share a branch. So we're going to say no because, and we're going to say, although they do have a shared common ancestor, they're in different families and the red panda is actually more closely related to the raccoon. That's our 3 marks for 3 points there. So is the mark scheme for question 1. So like we said, similar organisms, or you can say organisms with similar features for the first mark of the definition of a species, but you need to make sure you've said that. You can't just say a group of organisms. That will not get you the mark there. And they can reproduce and produce fertile offspring. You can say breed or mate instead of reproduce, but you need to talk about there being fertile offspring. Then we've got the groups within groups, or an idea of larger groups at the top and smaller groups at the bottom. Like I said, big groups divided into smaller groups, or smaller and smaller groups is fine, but that's the idea for Mark 1, and then Mark 2 is there's no overlap. Brown bear was the closest relative of the polar bear because they're on the same branch. There isn't anything else you can say for 1B. And then just saying no for whether there is evidence on the tree to support the conclusion doesn't get you a mark, because obviously yes or no would be a 50-50 choice, and that's not what we're after here. We're after for the explanation. So giving a statement and the disagreement no scores 2 marks. So giving one of these statements and no will give you 2 out of 3, and then getting all 3, so no and all 3 of these statements, gets you 3 marks. So you need to say they do share a common ancestor, which is probably the one that people will miss out the most. So we're kind of saying yes and no. So yes, although they do share a common ancestor, but the red panda is in a different family. So saying that it's in a different family and it's more closely related to the raccoon. That's really what we're looking for. But they do obviously share a common ancestor, so there is a positive to the fact that they are sort of related but not closely related. Okay, so question 2. Organisms and their interactions with habitats depend a lot on the size and surface area they occupy. Describe the relationship between size and surface area to volume ratio. Okay, so this is, again, kind of just a factual recall question. It should be a fact that we know or we've learned about the interactions between size and surface area to volume ratio. When it says relationship or describe a relationship, it's really just asking you how does one thing affect the other. So sometimes that's to do with the graph, and sometimes it's just to do with sort of saying how as one thing changes, something else changes. So in this case, it's as size increases, what happens to surface area. So hopefully we know that as size increases, surface area surface area to volume ratio, decreases. And you can write it as SA to V in terms of the little ratio sign, that's fine. People will know what you mean by that. Part B. Size is genuinely proportional to the mass of an organism. The range of sizes of land mammals and marine mammals is shown on the graph below. So it's kind of a sketch. There's no numbers on this graph, but that's okay. So marine mammals, just to make sure we're happy with what that means, it just means ones that are living in the ocean versus ones that live on land. And obviously we think about mammals, so things like whales, seals, the kind of big organisms that we know live in the sea that are mammals, otters, things like that. So we've got a range of sizes. So frequency on the y-axis really means here like how many individuals of that size are there. And so we've got mass along the bottom. So it's getting us to think about size. And then we're going to have to link that at some point to surface area to volume ratio, because that was what was in the first part of the question. So this is going to be trying to link those things together. So what other patterns can we see? So obviously we've got mass on the bottom. So smallest towards the left, largest towards the right. So there's a high frequency, or there's the most or the mean number of land mammals is kind of quite small. It's towards the small end, whereas the most or the mean or the largest frequency of marine mammals is somewhere kind of middling mass. And then the biggest mammal that we have in terms of size is a marine mammal, and the smallest mammal that we have is a land mammal, and there is an overlap in the middle between them. So they're not completely separate distributions, there is an overlap in the middle as well. So it wants us to use the graph to compare the size distributions of the two groups of mammals. Okay, so we're basically just going to see basically those patterns that we just started talking about, those distributions, the differences between them. We're going to start comparing them. Normally compare means similarities and differences. I'd say mostly here you're going to talk about differences, but like we said, there is that overlap. So that means that kind of some of the largest land mammals overlap in size with some of the smaller marine mammals. But there's lots of things to talk about here, and we can talk about the range, kind of the differences in the size, the differences in the means, and we've got only 2 marks for this question. So I would say pick one of the bigger differences. Obviously, clearly, the majority of land mammals are smaller, or most land mammals are smaller than marine mammals, because that's where the averages are. Whereas we can also say, sort of like a similarity, there is this overlap. So that's what I'm going to focus on, is talking about kind of the extreme difference between the average size and then also the overlap. Okay, so my first point is that on average, land mammals are smaller than marine mammals. And then my second point is that there is an overlap between the largest land mammals and the smallest marine mammals. And there's my 2 points. And I don't have space enough to squish that in the bottom, so that's what I would write, though, in that gap. Okay, so part 2. This is where we're going to need to link the 2 things that we've just been talking about together. So use your knowledge of surface area to volume ratio and temperature to explain in detail one reason why the average marine mammal size is different to land mammals. So it's 3 marks, but we're only talking about one reason. So this is the kind of one reason why we are explaining the difference in size. We need to link our knowledge of surface area to volume ratio, which it got us to repeat at the start. So the decrease when large, so the fact that larger animals have smaller surface area to volume ratio, or swapped around, and temperature. So thinking about surface area to volume ratio and temperature, well, normally we talk about diffusion of heat. So obviously, if you've got a small surface area to volume ratio versus a large surface area to volume ratio, the way that heat travels through the body is going to be different for those. So that's where we're going to bring in our link between temperature and surface area to volume ratio. And then it wants the reason for the difference, and we know that the difference is that marine mammals are on average bigger. So why, to do with temperature and surface area to volume ratio, are marine mammals on average bigger? Okay, so I've got 2 cubes here to represent a small animal and a big animal. So my large animal has a small surface area to volume ratio, and my small animal has a big surface area to volume ratio. In my small surface area to volume ratio, my larger organism, it takes a lot longer for heat to diffuse, to dissipate through and be lost from the surface area of the animal, because there's a smaller surface area compared to the large volume. Whereas in my small organism, we've got quite a short distance between the centre and the external surfaces, so it loses heat faster. So why would this be important for marine animals to avoid losing heat? Well, if you have ever been in the sea, in most places, it's incredibly cold. The sea is a very cold environment. It conducts heat better as well than in air, so you lose heat quicker in the sea as well. So if marine animals were smaller, they'd have a larger surface area to volume ratio and they would lose heat faster, which would mean they would be too cold. So smaller animals have a larger surface area to volume ratio, so they lose heat faster. Now we need to think about why. So we say water is cold, or water is a cold environment. We still don't have a why. So we need a link between the idea that water is cold and that if you're small, you're going to lose heat faster. Why is that important? Why would that mean that mammals have evolved to be larger if they're living in a marine environment? We need a why. We still haven't linked those together. We still haven't said enough with 3 points. So why do they need to be larger? Well, they're mammals. What are mammals? They are warm-blooded. They need to maintain their body temperature. Or if they were not large enough, they would not be able to maintain their body temperature, and then they would be too cold and they would die. So it's about maintaining body temperature, not losing heat because you will die, and you need to maintain your body temperature because of enzymes and all of those things. So marine mammals need to be large to maintain their body temperature. Part C. A new study indicates that there has been a large decrease in the mean and maximum land mammal size since the late Pleistocene era, which is roughly 125,000 years ago. This decrease was found together with the increased geographical spread of humans around the world. suggest a reason for this decrease. Okay, so you'll know hopefully that sometimes we add these extra questions onto the end of maybe a long sequence of questions that's related, but it is suggest at the start. So that means you need to take information from the question, mostly all the questions you've answered up until this point, and the question information itself, and use your judgment, your common sense, your scientific knowledge to try and suggest an answer for this. It will not be something you have learned. It will not be a piece of content that you have been taught. This is you interpreting data, interpreting information, making links based on what scientific knowledge you have up until this point. So we need to find a link, or what we think might be a link, between a decrease in large mammals and a decrease in the average size of mammals. So like a decrease in the fact that they get smaller because there's fewer large mammals, but also how many there are as well, and an increase in humans spreading out across the world. What link could we make between those? Well, basically it's asking you to think about what humans could have had on the impact of a distribution or species numbers. Clearly, humans hunt organisms, and they would have continued to hunt organisms if they spread out around the geographical areas. They would have hunted large animals because they would have been maybe slower moving or easier to spot, easier to take down, something that doesn't run very fast. Often large mammals are herbivores as well, so they're not carnivores, things like that. So the idea that the humans hunted more of them and killed more of them, and therefore the average size decreased because we hunted things like the giant woolly mammoth into extinction. So that is the kind of answer that they're trying to get you to get out of that point. But as I said, not something we should have necessarily learned, but we have used our knowledge and our common sense to come up with an answer there. Okay, so the mark scheme for question 2, we said as size increases, the surface area to volume ratio decreases. You can say it the other way around. So as size decreases, the surface area to volume ratio increases. That's absolutely fine. We've got some options for our kind of pattern spotting and talking about the differences between those graphs. So the idea that the average mass of land mammals is smaller, which is what we said, or you can say there's more number of smaller land mammals than marine mammals, so there's more smaller land ones than marine ones, or the other opposite end of the spectrum, that the average mass of marine ones is larger. That's kind of 3 points in one, but they all kind of are around the same point, so that's why you can only get 1 point for any of those. The fact that the largest marine mammals are larger than the largest land mammals, so there's a difference there in the largest of each one. And similarly, the smallest land mammals are smaller than the smallest marine mammals, so there's a more small ones as well. And then that there's an overlap, which is what we talked about in size, between the largest land mammals and the smallest marine mammals. That is the one that we talked about. So any two of those, but obviously within those sort of bullet points, gets you 2 marks. Then the 3 points, as we said, so small animals have a larger surface area to volume, so they lose heat faster. Or the other way around, if you have a smaller one, you're going to lose heat slower. Water is a cold environment, or water is a good heat conductor, or they live in colder habitat. Any of those is fine. So marine mammals must be large or have a small surface area to avoid heat loss. So we didn't really necessarily talk about that part, but you can get a point for just saying, well, if you are large, then you're going to lose heat less. And then if they were too small, they would lose too much heat, or they would not be able to maintain their body temperature. That's the why. But any 3, because there's technically 4 points here, any 3 from those gets us marks. And then last one, increased hunting. If you said killing, you said a threat to large mammals, any of those idea that we were a threat, we were going to kill them, we were going to hunt them, we have reduced their populations, the large size populations, in some way. Question 3. Hemoglobin is an example of a protein that has a quaternary structure. Explain what is meant by a quaternary structure of a protein. Okay, so we've got that sort of phrase again, explain what is meant by. So it just means define, really, in this kind of question type. So basically define quaternary structure of a protein. So quaternary structure, hopefully we remember, we have primary, secondary, tertiary, and then quaternary is the last one, where we have multiple polypeptide chains joined together. B. Explain why oxygen leaves hemoglobin when red blood cells reach respiring tissues. So What happens to the blood when we get to a respiring tissue? So respiring tissue here basically just means tissue cells that are doing respiration, so they're taking up oxygen and they're releasing carbon dioxide. So what happens when the blood or red blood cells reach these tissues? What happens to cause the oxygen to leave the hemoglobin and go into the tissues as opposed to staying bound to the hemoglobin? What causes that change, that dissociation? Okay, so if we're using up oxygen, we need to say how we're getting the oxygen to do that. But we're releasing carbon dioxide at these respiring tissues. That's going to be dissolving in the blood plasma and forming carbonic acid, which lowers the pH by increasing the number of H+ ions dissolved in the plasma, and that is what causes the oxygen to dissociate from the hemoglobin. So we get oxygen dissociation, then the oxygen obviously can diffuse into the tissues. This seems like a lot, but it's a 3-mark question, so we need to say enough to get 3 marks here, and we can't just repeat too much of what's been said in the question. So we need to say there's increased carbon dioxide due to the respiration happening, so we understand that respiration means more carbon dioxide. That's 1 mark. Then we've got to say what that does, so that creates carbonic acid, which lowers the pH, and then that is what causes the oxygen to dissociate. Okay, so part C. Some long-distance runners spend part of their time training at a high-altitude camps. Some data was taken before and after their time training there. This data can be seen in the table below. Okay, so they went and trained at high altitude. So my first thinking when we see high altitude is the fact that that's got low partial pressure of oxygen or low oxygen concentration in the air. And so they sort of had something measured before they went and then after they trained at this kind of low oxygen camp. And then we've got a table, so we've got the before the high altitude, after the high altitude, we've got the red blood cell count, and we've got the oxygen uptake. Now, if we have a look at the numbers, there's an increase in both. So there's an increase in red blood cell count, increase in oxygen uptake. So part 1 says, compared to low altitudes, why might runners find it more difficult to exercise in the same way at the high altitude training camp? So we are comparing them, basically looking at those 2 sets of numbers, so before and after. So at low altitudes would be before. Why might they find it more difficult to exercise at the same way? So same sort of level of exercise would require the same amount of oxygen to the respiring tissues, but if we're at high altitude, what makes it harder? Well, we don't have as much oxygen in the atmosphere. We have that low partial pressure of oxygen or low oxygen concentration, so that means that they're going to have to work harder. They can't get the same amount of oxygen to their tissues breathing normally, or as they would at low altitude. So we're going to say that obviously at high altitude, there's low partial pressures of oxygen, which means they're not able to get enough oxygen. And we're talking about exercise, so why do they need oxygen? Well, they need oxygen to do respiration. So they can't get enough oxygen to their muscle cells to do respiration, which will then means they'll find the exercise harder than they would at normal low-level altitudes. Calculate the percentage increase of red blood cells the athlete had from attending the camp. Okay, so this is a maths question. It's just using some data to do a percentage increase, which is just a percentage change calculation. So for any percentage change, whether it's increase or decrease, you work it out in the same way. We need the change, or the difference, divided by what the number was at the start, times 100. So we need to show our working, and because we've got the numbers as 4.32 and 6.51, and they're both times 10 to the 12, we can just do it with the numbers in front of the standard form, because the standard form isn't necessarily important here. So 6.51 take away 4.32 gives us the difference, or the increase, and then we're dividing that by the original number, which is 4.32, because that was the before number, times 100 gives us 50.69%. And we'll just leave it like that. That's enough. That's fine. Finally, part 3. After months of training at high altitudes, the athletes have an advantage when they return to compete in races at sea level. Use the table to explain why. So we have to use the table. So we've got to use kind of what we've been given, the information we've been given in the table. So that's going to give us our reasoning. So we've got obviously more red blood cells and therefore more oxygen uptake. So we needed to say why that is an advantage when doing exercise back down at sea level. So what advantage does that give them? So we're going to talk about the difference that they've got, or the advantage they've got is that they have more red blood cells, and then link that to why that is an advantage Well, that means they get more oxygen, but we need a link between those two. You can't just talk about more red blood cells equals more oxygen. Why does that equal more oxygen? So it's an advantage because that means they have more hemoglobin that can bind the oxygen. That's the kind of point here. They want us to make that link. If we're just saying, oh, they've got more red blood cells and therefore their maximum oxygen uptake is increased, that's just regurgitating what we see in the table. We need to make a science link there to get the marks. So more red blood cells means more hemoglobin, which means they can get more oxygen to their respiring muscles. Remember, we're talking about exercise. We need to link why that's an advantage. They have more oxygen transport, which can then get to their muscles, so they can do more respiration, which helps them when they are doing exercise. So more red blood cells, so more hemoglobin to bind more oxygen and transport to muscle cells. The word more is really important here because we're saying what's their advantage. So if you have more red blood cells, that means more of this, and that's why it's an advantage to them. So the mark scheme for question 3. Made of more than one or multiple polypeptide chains, just make sure you've talked about being joined together, okay? We've got sort of our 4 points potential for 3B, so any 3 from these. So with the low pH or more H+ ions, increased carbon dioxide due to increased respiration. We said that the other way around. That's absolutely fine. Increased dissociation of hemoglobin. I would say here you absolutely have to have that word because it does say oxygen leaves hemoglobin in the question. So really we need to understand that obviously it dissociates, it drops it, it unbinds, whatever you want to say, but dissociation is the word we need there. And then the oxygen diffuses from the red blood cells to the tissues. For C1, we have to say partial pressure of oxygen. You can't get away with not saying that. It needs, if it's underlined, you need to say that word to get the mark for that first one. And then for the second one, not able to get enough oxygen to maintain respiration, or not able to get enough oxygen to get to the muscle cells. So you need to link here this idea of oxygen and respiration, or oxygen and getting it to the muscle cells. What do we need the oxygen for? That's what has to be in there to get that mark for C1. Then you've got your 1 mark for the percentage calculation. It's only 1 mark, so you can't get any extra method marks or anything like that. You either get that answer or you don't. Then we've got more red blood cells, which is what we interpreted from the table for C3, and then more hemoglobin available. So that's the most important part here. You can't say more red blood cells, more oxygen. You need the link between why more red blood cells gives you more oxygen transport, and it's because there's more hemoglobin available to bind the oxygen. So you need both parts there. Question 4. Before beginning training for a race, an athlete went through a medical examination. The graph shows the blood pressure and volume changes in the left ventricle and the heart sounds measured during 1 complete heartbeat. Okay, so we've got 3 kind of graphs here almost in one. We've got time in seconds along the bottom, we've got heart sounds, we've got the volume of the left ventricle, so the volume of blood in the left ventricle, and then blood pressure.
So we're going to have to think about how all of these things are linked together. They're clearly layered on top of each other, so it's kind of like a cardiac cycle graph, if you've seen one of those before, which is kind of looks more similar to the blood pressure graph normally. But we've got all of these things lined up so that clearly things are happening at like 0.4 and 0.6 seconds with the sounds, and then those lines continue up so we can kind of track what's happening at each point. Because I kind of know and remember some things about the cardiac cycle graph, I might want to just annotate some things that I know are happening. So obviously here we've got the 4 points at which there's sort of crossover, or there's normally things happening. We know there's something definitely happening at these 2 points. This is obviously the lub sound, and then this is the dub sound of the heart, sounds the heart makes when it beats. And so this is caused by the AV valves, and this is caused by the semilunar valves, which means that this is something to do with the AV valves, and this is to do with the semilunar valves. And normally we are talking about them closing, which is what makes that sound. So then at B, then we've got things happening in terms of what's opening and closing as well. We can talk about that thinking based on the pressure. So clearly the pressure is increasing, and that's because the ventricles are contracting, which is what's causing the pressure to increase. This is also the AV valve as well, and this is the semilunar valve as well. Okay, so these are just kind of the annotations I'd make in terms of just looking and trying to figure out and understand this graphic because there's a lot going on here, and we'll have a look at what the questions want us to do and figure out those answers as well using the annotations. Part A.
The heart sounds were recorded using a phonocardiogram. The sound made at 0.4 seconds is caused by closing of the AV valves. What makes the sound labelled C? Okay, so basically, what are the two heart sounds? One, they've told us the first sound is the closing of the AV valves. So what makes the second sound? Well, it's the closing of the other set of valves. So the AV valves closing gives us the kind of lub sound, and then the dub sound comes from the closing of the semilunar valves. Now that makes sense here because you can see the pressure is dropped right down in the ventricles, and that's because the pressure in the aorta is then higher than in the ventricles, so that semilunar valve closes. Part 2, what causes the AV valves to shut, which makes the first sound? Okay, so this is asking for the reason why. Why do the AV valves shut? Again, so we said the semilunar valves just now, we said they shut because the atrial pressure, the aortic pressure, becomes higher than the ventricular pressure, so that causes them to shut. This is the same thing. Any valve is going to close when there is a change or a difference in pressure. So in this case, we've got lower atrial pressure and higher ventricular pressure. So the pressure in the ventricles is higher than in the atrium, and so that's what causes the closing of the valves. So the pressure in the ventricles is higher than in the atria. Using evidence from the graph, explain what is happening in the heart at point B. So point B is up here, so it is after the AV valves have closed. We've just said that the pressure in the ventricles is higher than the atria, which is why the AV valves have closed. We can also see that there is a big decrease in the volume of the left ventricle. So the volume of the ventricle is getting smaller. We said the pressure in the ventricles is getting higher, which you can see because it does increase above B. So that tells us that the ventricles are contracting. So what's happening at point B is the ventricles are contracting, and the reason we know that is because the pressure continues to increase, but also the volume of the ventricle decreases. So the volume is getting smaller, the pressure is increasing. That's because the ventricles are contracting. Now, I'm putting all of the detail here because it says using evidence from the graph. So it's 2 marks. So we're likely to get 1 mark for saying the ventricles are contracting, but we need to explain using evidence from the graph. So our reasoning needs to be from the graph. So because the volume of the ventricles is decreasing, I would say, and also the pressure is increasing. And then you've covered yourself with both bases. Okay, so part C. In this person, the stroke volume is 70 centimetres cubed. Calculate the cardiac output. Okay, so this is one of those equations that we have to know. There's not very many that we need to know for biology, but this is one of them. We should be able to know how to calculate cardiac output, and it wants us to show our working. So I've brought over the graph because we're going to need it, because we are going to need to calculate the heart rate, because cardiac output is the stroke volume multiplied by the heart rate. And the heart rate is always in beats per minute, and we've been given the stroke volume as 70 centimetres cubed. So we've got the time in seconds on the bottom of our graph here. So we're going to use this to calculate heart rate. It said also in the question that this was 1 complete heartbeat. So 1 heartbeat is 0.8 seconds. So we know that that's the time for 1 heartbeat. So then we just need to divide that by 60 seconds, which is a minute, to find out how many beats per minute there are. And obviously we're going to show our working, so we're going to show what we're doing from the beginning. I would also write out the equation that we're using as well. It's probably the best idea to show what you're doing. So 60 seconds divided by 0.8 gives us 75. So our heart rate is 75 beats per minute. We're going to multiply that by 70, and that's going to give us the stroke volume. So it's 5,250 centimetres cubed per minute. Centimetres cubed per minute is important here because there are no units on this question, so we're going to have to make sure we know the units ourselves. Obviously we're sort of saying per minute, so centimetres cubed of volume per each minute. That's where we get that unit from. So part D, after 2 months of completing a training regime to prepare for a race, the athlete was re-examined. They were found to have the same cardiac output but a lower resting heart rate than before. Explain what effect the training has had to cause on this change. Okay, so how can this happen basically? So they've done some training and they were found to have the same cardiac output, but even though they had the same cardiac output, it was their resting heart rate that was lower. So if we look at our equation again, we've got cardiac output equals stroke volume times heart rate. This is saying then that we've got the same cardiac output because for a lower heart rate. So if our heart rate has decreased, our stroke volume must have increased in order to make that number the same. That's the only way that can happen. So we can say the stroke volume has increased, that's one mark, but that's not the why. That's not the reason why. So the stroke volume, we need to know what the stroke volume is. Like, what does that word stand for? It's the volume of blood pumped out of the heart. It's normally measured as the volume of blood coming out of just the left ventricle, but it's being pushed out by the ventricles. So it's how strong that ventricular contraction is, or the size or volume of the ventricles is going to determine how much blood is pumped out. So if our stroke volume has increased, it's for a couple of reasons. Either we've made the ventricle bigger, increased in size, increased in volume, so more blood is being pushed out each time, or the force of contraction is stronger, so more blood is physically being pushed out each time. Think about what they've said. They're doing training. Any exercise is training and improving the strength of muscles. That includes your heart muscle. There's lots of ways we can say this, but the way I always like to think about it is if you're doing training and exercise, you are strengthening your heart muscle. So that means the heart muscle is stronger, it's strengthened, and therefore it can contract with more force and therefore push more blood out every time the heart beats, which is what increases the stroke volume. We'll look at the mark scheme because there's lots of ways you can say this. But I've gone for talking about the heart muscle being strengthened, so more blood is being pumped out with every contraction. Okay, so mark scheme for question 4. Closing of the semilunar valves is what we put. You can say pulmonary and aortic valves if you've learnt those instead. When the pressure of the ventricles is higher than in the atria, just make sure you've said higher and you've got that clear difference there with A2. For B, you can say systole instead of contraction, that's fine. And you, as we said, you can either say because the pressure is increasing or because the volume is decreasing. Either of those is evidence from the graph, so that's fine for B. You get the correct answer, you get 3 marks for the calculation. If you didn't get the answer for whatever reason, then if you wrote down the equation like I said, you can have a mark for that. And if you showed how to work out beats per minute, you can have a mark for that. This is why we always show our working, just in case. 4D, increase in volume or size of ventricles, as I said, or increase in volume or size of the heart, or increase in strength of the heart muscle, which is what we said, or increasing the strength of contraction. Any of those will get you the first mark for part D, and then you need to say so more blood leaves the heart in each contraction, or just say that has increased the stroke volume, because we obviously worked out that the stroke volume had to increase in order for our cardiac output to be the same. So you just need to say that that's what happens and then say why, using one of the options for the first mark. Okay, so question 5. The diagram shows the influenza virus. Okay, so we've got a diagram. Influenza, obviously is just short, the short version of that is flu. So we want us to identify structures A and B. So you may have learned different virus structures in different formats, in different diagrams, that they can look different, but we should know kind of some of the basic structures. So the attachment proteins are A, they are the kind of spikes that stick out around the edge on a virus that looks like this. And then B is surrounding what we can assume is the DNA or the RNA or the genetic material. So B is protecting that. It is a protein coat, or you may call it a capsid. Be careful, it is not the capsule, that is in bacteria. So you can call it the capsid or protein coat, and that is surrounding and sort of it protects the genetic information. What is the function of C? So it's not asking us what it is, it's just saying what does it do? Well, any nucleic acid is going to contain genetic information. So it's carrying or contains the genetic information. Some viruses, including influenza, have enzymes which hydrolyze structures in the cell surface membrane of human cells. In the diagram, these are labelled D. Part 1 says, suggest how these enzymes are useful to the virus. So this is a suggest question, which means you need to use information from the question and your own knowledge, but in this case, it is knowledge of sort of viruses and how they work. Sometimes it'll be knowledge like common sense knowledge or things that you might have picked up from the course and not something you know specifically, but we do know how viruses work and it's something that we have been taught. So we basically just need to think about why would it be useful for a virus to be able to break down or break open the cell membrane. So we know that as a virus, they replicate or need to be inside cells in order to survive. So they have to get inside in order to use the cell's machinery to replicate, and then they have to get out again to go and infect more cells. So these enzymes are useful in two ways. They could either help the viruses get into the cells, or once made, because remember these viruses were made entirely inside the cell, they could help the viruses to escape and get out, break out of the cell in order to then go and infect more cells. We don't know for sure which one it is, but it could be either of those, and we need to say something that's worth 2 marks, so there we go. Part 2. New drugs to treat influenza are being tested. One type is a non-competitive inhibitor of the enzymes found at D. Explain how this type of drug would act to treat influenza in a patient. Okay, so although this is dressed up as an immune system flu drug question, what it's actually asking is just, how does a non-competitive enzyme inhibitor work? That's it for 3 marks. What will this non-competitive inhibitor of this enzyme do? And that's all that we need to say. So hopefully we've learned the process and steps that come along with what an actual non-competitive inhibitor of an enzyme does, and we just need to write those out. Just being careful about our keywords and making sure we're saying everything we need to say. So it doesn't attach to the active site, it attaches to somewhere else on the enzyme away from the active site. Sometimes we call this the allosteric site. Once it binds, it causes the shape of the active site to change, which means that the enzyme is no longer complementary to the substrate, which means it can't form enzyme substrate complexes anymore. And if it can't do that, then it can't break open the cell membrane, and therefore the virus can't even get in or get out, depending on which use it's for. But that last point we don't really need here, because it's just saying how would this drug work. So I've written this out long form. You can use the kind of short form we did, but you need to make sure we include all the right words. So it attaches to a site away from the active site and causes the active site to change shape so that it is no longer complementary to the substrate. This means no ES complex can be formed. Or you can say the substrate cannot bind. Either is fine, full sentences, or those bullet points that we did on the right-hand side. So the mark scheme for question 5, pretty straightforward. We've got our attachment protein, you've got capsid, capsomere, or protein coat for B for 5A. So any of those is fine. I didn't mention capsomere, but if you put that, you can have a mark. 5B carries genetic information. You need that in your answer, really. That's the only kind of really acceptable point there. We've got this idea of being released from infected cells, also a virus can enter cells. You can have those two as long as you talk about breaking through the cell membrane and then giving those two as points, like we did. Other than the active site, we said you can have allosteric site for C2, changes shape of the active site, or you can say changes the tertiary structure of the enzyme. I would always go for active site or say both, because the kind of marks for these change, and realistically I've seen active site required for these marks more than I've seen tertiary structure on its own. So say both, or just say active site, but don't just say tertiary structure. I would. And then you need the word complementary to get the last mark. You need it. So you can't get away with not saying that word, and we shouldn't be avoiding that word if we're writing a question about enzymes, full stop. We should always be using the word complementary. And then you need that kind of joining sort of point there where it's not complementary. Okay, so what? So there's less or no substrate binding, or less or no ES complexes forming. Question 6. The diagram below shows the life cycle of a slime mold, Fuligoseptica. These organisms share features with fungal cells but are classed as protists after it was discovered that they do not contain cell walls. Okay, so we've got a life cycle of a protist. Protists are like malaria. We don't really learn a lot about protists, but that's fine because we'll probably have all the information we need. They don't get to the cell walls, that's a good thing to know. And then we've got this life cycle diagram. So hopefully you may have seen some of these life cycle diagrams. Often it's to give you alternative kind of views of mitosis and meiosis. So they're different from kind of the normal life cycle we think of in terms of human sexual reproduction or mitosis in terms of like cloning and growth. So I would always say when faced with one of these diagrams, what we need to do is look for clues that tells us where mitosis and meiosis might be happening or what types of cells we're dealing with. So N and 2N is a way of describing the number of like chromosomes. So N and 2N, you can see N, if N was doubled, then it would be 2N. So 2N is often a code for diploid cell, so double the number of chromosomes or the full set of chromosomes. And then N would be half the number of chromosomes, so it would be a haploid cell. This makes sense because if the spores are haploid, you can see we've got a spore germinates and it produces these cells, either flagellate cells or amoeboid cells, and then these cells fuse. So the cells fuse and then we get a diploid organism. So that's kind of like sexual reproduction. And so if the spores are haploid, then they must be more like gametes. And because they're behaving like gametes, there's two types, and then they fuse and then they create a diploid cell. So that I think is as much as we need right now to understand the diagram. And if we need to, we'll come back to it once we've figured out what the questions are asking us. So part A says, if the slime molds had the same cell walls as fungi, what substance would they be made of? Okay, so this is just a, do you know what the cell walls of fungi are made of? Chitin. Really cool question, it's just something we have to know. B says, which letter on the diagram shows where meiosis occurs in the life cycle? Okay, so this is a common question for questions like this, where they're talking about life cycles. They want you to either spot or identify where mitosis has happened or where meiosis has happened. So if we think about what is meiosis, it's the cell division process that produces gametes or produces haploid cells. So we said here where the spores are, N, that must be haploid, like gametes. That means whatever process is going on at C to create the spores must be meiosis. So our answer is C. Part C, explain the importance of meiosis in the life cycle and development of this organism. Again, a common question about meiosis, and really wherever we look at meiosis, whatever type of question you get, it's really just asking like, why is meiosis important for survival? What's the point of meiosis? Why is it important that organisms can do it? And sort of an easy way to think about it sometimes to think about what would happen to an organism that couldn't do meiosis ever and could only reproduce asexually by mitosis. What would they be missing out on? What does meiosis provide that helps this organism? Why sometimes will they need to do sexual reproduction versus the asexual reproduction? So the main thing is it increases genetic variation because obviously it involves crossing over and mixing of genes from two different cells. So by increasing the genetic information, the genetic variation, you're more able or more likely to be able to adapt to a changing environment than if you are just clones and always a clone. If you can mix genes, then you can get a genetic variation, increase that, and therefore you may be more likely to be able to cope if the environment changes. The reason we can do that, we can mix genes from different cells, is because meiosis makes haploid cells, which can then come together and fuse to make a whole cell with all of the genetic information. If that didn't happen, if we didn't make haploid cells that then could fuse to make a whole new organism with the right number of chromosomes, then we wouldn't be able to maintain our chromosome number, and we'd end up having to double and double and double and double the genome every time, which is just not possible. So 3 marks, we need to say kind of 3 things here. So meiosis produces haploid cells. So even if you weren't sure, just describing what happens in meiosis is probably going to get you marks here if you really weren't sure how to answer this question. So meiosis produces haploid cells which can fuse to become a diploid cell or a diploid organism. This increases the genetic variation in the cells or in the organism, which allows or makes the organism more able to adapt. Can't say it is adapted or can't say it becomes adapted, because it doesn't. That's not what an adaptation is. Thinking about natural selection, it just means that by having increased genetic variation, it's more able to or likely to be able to adapt to a changing environment. The amoeba stage is free living and feeds on bacteria. Describe how the amoeba could eat a bacterium. So describe how, as always, is our steps in a process question. So it wants to see you describe the steps that are going to occur in this process. And it's asking, how could a single cell, like an amoeba, eat, that means like take inside and digest, a bacterium? The way it could do that is through phagocytosis. This is clearly just a phagocytosis question dressed up in a different way. It's asking, do you understand how a single cell could eat, for want of a better word, but digest a bacterium? So it's the same process, and then it would come along, it would detect the bacteria, it would engulf it, then it would put it inside, as it engulfs it, it would put it inside a vacuole or a phagosome. Then it would have lysozymes, a lysosome containing lysozymes or other enzymes, digestive enzymes. Those two would fuse, and then the bacteria would be digested, and then those molecules would be absorbed rather than being ejected, like in phagocytosis. So that's what we'd say. Go through these kind of 4 bullet points. So engulfing, forming a phagosome, the lysosome fuses, and the lysosome contains enzymes which digest the bacteria. That's it. So the amoeba engulfs the bacterium. It encloses it in a vacuole called a phagosome. A lysosome containing enzymes fuses with the phagosome, and the bacteria is digested. Do not use the word eats here. You need to use the word digested because you're kind of showing that you understand that this is a digestion process. You're going to use enzymes to break down the bacterium into small molecules which can be absorbed. So I would definitely use that word. You can't use the word eats anyway, throwing it back from the question. Okay, so the mark scheme for question 6. We've obviously got Kitten and then C for part B. There's not really anything else to say about those, they're quite easy. So then you can any 3 from this list of 4, which I think we said all of them. So meiosis halves the chromosome number or produces haploid cells, we said. When gametes fuse at fertilization, the diploid number is restored. We said forms a diploid organism, which is fine. Introduces genetic variation. You can say how and you get marks for that, so that it causes independent assortment or crossing over. But there are slashes, so you don't need to say all of that, just genetic variation is fine. And then more able to adapt to environmental conditions. So any 3 from these got you 3 marks, you don't have to say all of them. And then 4 from any of these. So there's again quite a lot that you could get marks for here. So you could say that the amoeba is attracted by a substance or it recognises the bacteria in a similar way to how phagocytes obviously detect pathogens. It's engulfed or ingested, either is fine. Enclosed in a vacuole, a vesicle, or a phagosome, also fine. The phagosome fuses or joins with the lysosome. The lysosome contains enzymes, and the bacterium is digested, or it's hydrolyzed and absorbed. So not eat or broken down, you can't have that because again eat was in the question and you need to say hydrolyzed or digested, you can't just say broken down for the last mark. Instead of saying fuses or joins with the lysosome, you can say it forms a phagolysosome, that's fine. If you just described the engulfing, so got like described it wrapping around the cell, then that's also acceptable for the talking about engulfing point. But you just need any 4 of these.
Okay, next question. So, scientists wanted to investigate stomatal density on leaves from 2 plant species from the genus Aster. Aster amellus grows in mountainous regions, and Aster tripolium is a halophyte and adapted to live in salt marshes. In order to view the stomata, the scientists removed small thin pieces of the lower epidermis from leaves of each species and mounted them on microscope slides. Now, if you haven't heard of a halophyte, it means salt-loving. Literally anything prefix halo is to do with salt, and halophyte,
that idea of being a phyte means you love something or are kind of adapted to it in this case. So it's normally plants that are adapted to live by the sea or in kind of salty soils. Now, if we're putting small, thin pieces of epidermis and we're looking at stomata, we're going to be using a light microscope. And so that's going to hopefully help us get in the right mindset for these questions, because it's clearly going to be a sort of microscopy question that's also related to stomata and plants. So the first question is part A, explain why it is important for the leaf tissue sample to be thin. Okay, well, there's a clue here. It's obviously to do with the light microscope. If we're using a light microscope, we need light to be able to pass through our sample in order to be able to see it. So if we have a really thin layer of tissue, we want it to be about a single layer of cells or not loads of layers of cells on top of each other, so the light can pass through it so that we can see it. Suggest how the scientists use their slide to find the number of stomata per centimetre squared. So this is a kind of method suggestion question. Say how they'd use it, a way of counting the number of stomata, how you'd calculate the area that number of stomata in, and then how you'd obviously multiply that up to a centimetre to get the number per centimetre squared. And also you need to do multiple counts and take a mean. If we know anything from things about counting small things like whether it's bacteria under the microscope, red blood cells under the microscope, whatever it is, if you're counting something in a microscopic sample, you need to do it multiple times and then take a mean of that to get the average number of stomata, in this case per centimetre cubed, so per centimetre squared even. So we're going to need to calculate our area of the field of view, or you could talk about calculating the area of the leaf tissue you're looking at and say you're going to count and measure all of it, but it seems unlikely to do. If you've got the circle that you're looking down and you can work out what area of the leaf that covers, then you know how many is in that field of view. You just move it along one, and then you can kind of repeat the process just by moving the viewfinder. So calculate the area of the field of view of the microscope using a calibrated eyepiece graticule and the formula pi r squared. So if you can measure, you know what the width in terms of, it's probably going to be micrometers or millimeters to start with, the width of your field of view. That gives you the diameter. Then you half that to get the radius. You can do pi r squared. It will tell you the area in actual numbers of your field of view. Count how many stomata are in there. Do that and take a mean, and then you'd have to just times it up to get it per centimetre. So count the number of stomata, repeat, calculate a mean. There we go. The stomata of both species close when the light is turned off. Explain the advantage of this to the plant. So realistically, if we're turning a light off, what is that representing? Well, that is representing nighttime, daylight is gone or light is gone. So when light's not available, why would the stomata close? And specifically, why would that be an advantage to the plant? Why would they want to close their stomata at nighttime? And realistically, it's to prevent water loss. So water can leave through the stomata. If you don't need your stomata open for gas exchange, then you'll close them to prevent water loss and to conserve water so that your plant does not dry out or lose turgor pressure or suffer from wilting, etc. So, but yeah, that's why. Okay, when light intensity is high, potassium ions are moved into the guard cells. Describe how the movement of potassium ions into the guard cells causes stomata to open. Okay, so this is really like an osmosis question. Do you understand how guard cells work? Do you understand how osmosis works and how those two would link together? So if you remember, the guard cells are kind of sort of sausage shaped when that stoma is open. And so they're only like that when they're turgid, so when they're swollen, full of water. And so in order to get them to be swollen or full of water or turgid, we need water to move into them by osmosis, which means you need to create a low water potential inside the guard cells for the water to move in. That makes sense. If potassium ions are pumped in there, I think they're actively transported in there, then you're going to be able to have a low water concentration inside the guard cells. So water will move in, plump them up, make them turgid, and they swell up unevenly. So that kind of middle cell wall part around the hole is rigid and stiff. It doesn't stretch, it doesn't elongate, whereas the outer cell wall is a bit more stretchy. So when they expand in this kind of banana shape. And so yeah, we're just going to need to describe the steps in the process about how we get to having this kind of sausage shape guard cell. Potassium ions lower the water potential inside the guard cells. This creates a water potential gradient,
So water moves into the guard cells by osmosis. Or you could say it creates a low water potential inside the guard cells, so water moves in by osmosis. And then they swell up or they become turgid unevenly, which causes the stoma to open. The scientist wanted to compare the stomatal density of the two species, give a null hypothesis for this investigation, and name a statistical test that would be appropriate to test the null hypothesis. Okay, so if we're comparing stomatal density, and we've already said how we'd work out the stomatal density, and that would be like the mean number of stomata per centimetre squared, then we're really going to be testing the difference between two means, aren't we? We're going to be testing the difference between two numbers, the mean stomatal density of each one. That means if we're testing the difference between two means, we're going to do a t-test. And not a paired t-test, just a normal unpaired t-test. And then the null hypothesis, hopefully you should know, but null meaning nothing, none. So the null hypothesis is always that there's going to be no difference. There's going to be no correlation. There's going to be no relationship. There will be no difference between the observed and expected. Whatever test you're doing, the null hypothesis is always a negative. So in this case, we're going to say there's no difference, or there will be no difference between the stomatal density between the two species. That's our null hypothesis. And we're going to test this using a t-test. Halophytes, like A. trypolium, can use sodium ions instead of potassium ions to regulate their stomatal opening. When AMLS plants are exposed to salt-polluted soil, the sodium caused the stomata to open. But afterwards, the stomata no longer responded to darkness or carbon dioxide levels. So just how this would eventually cause the death of AMLS. Okay, so this is a suggest question. It means we need to use our common sense and information that we've got from the question so far. So if they wouldn't respond to darkness or CO2, we've already been told that they close when the lights turn off. So even if you didn't sort of realise what that was about, if we're saying that they won't respond to it being dark or to high carbon dioxide levels, they wouldn't close, which means they would stay open. And it told us why would they close in the dark. That question we answered previously, we said to stop water loss. So if they won't close their stomata or can't close their stomata, so it means they stay open all the time, then how could that cause the death of a plant? Well, it would just die due to water loss. Water would keep evaporating from the leaves, and therefore we wouldn't be able to maintain water pressure or turgor pressure in the plant. Okay, so here's the mark scheme for that question. Mostly self-explanatory, just draw your attention to 22C. So you could have just said 2 marks like we said for saying the water is lost through stomata, so closing them prevents water loss. But it's really to maintain water content of the cells, maintain the turgor pressure, reduce wilting, stop it from becoming dehydrated. It's not about photosynthesis. So it's not about saying, well, if you close your stomata, you stop doing photosynthesis because it's nighttime. The point is they don't need to do photosynthesis in the nighttime, so they don't need their stomata to be open, but they close them to stop water being lost unnecessarily. So it's not about photosynthesis, this question. And obviously now we've done the last question, that makes sense because it's getting you to think about this idea of water loss to help you with that last part of the question. We've got 4 options for the marks, and so we've got other things that we can say. So the part we didn't really talk about for 22D is we didn't describe how the inner wall is less flexible. I talked about it, but I didn't put it in my answer, but you can, and that would have got you a mark as well. Obviously also talking about decreasing the concentration of water, decreasing the water potential. There's loads of ways you can say it. Just make sure you've said it correctly. So you've talked about water concentration, not concentration. You've talked about water potential, or you've talked about something making it more concentrated or less dilute. It's just being really specific and making it clear. What do the potassium ions do when they move into the cells? How do they create that gradient, and what is the gradient? Where is the high water concentration? Where is the low water concentration? We've got another option for the null hypothesis, so you can say the stomatal density of both plants would be the same or equal. That's the same as saying there's no difference, so if you've worded it that way, that's fine.
Question 8. Now this is the first of our 2 long answer questions at the end of the paper, which is always what happens at the end of AS Paper 2. So these are one of the ones where it's like process, easy mark descriptions or explanations, but they're process steps. So explain what is meant by stabilising selection and describe the circumstances under which it takes place for 5 marks. Okay, I would say these questions are really nice. I really like them because they're not really application questions. They're just tell me what something is and what you know about it for 5 marks. So what is meant by stabilising selection? That's that phrase again. It means define it. So talk to me about what it is. It wants you to describe the circumstances under which it takes place. So steps in the process. What is it? What happens? When does it happen? Okay, so I've put this little graph in because hopefully this little graphic will help us. This is kind of what I would do. I would sketch a little graph to remind me what the stabilising selection graph looks like, and then just annotate it with the things I'm going to need to mention. So this is the kind of standard normal bell curve for distribution. So we've got mean in the middle, we've got the range. Then if we think about what does stabilising selection do to this bell curve, it makes it narrower and taller. I could have gone slightly taller there, to be honest, but it's fine to show that it's narrower. The mean, the peak, is still in the same place. The mean does not change. The extremes have changed. So we've really narrowed the range down and we've got much less of the extremes of both ends. So really, for example, let's think about this maybe being about size, or the example you may have learned was birth weight. So the extreme birth weights, really tiny babies and really heavy babies, are selected against, and we narrow the mean down until the majority of the population has the phenotype of being within that certain weight range when they're born, because 2 small babies won't survive, 2 large babies will probably die in childbirth potentially. So we get this kind of narrowing of the phenotypes. The mean stays the same, and that becomes the most popular or the most common phenotype in the population. And it's because it's the most adapted or the best adapted. Whatever that phenotype is that's around the mean is best for the environment. You're more likely to survive and reproduce if you have this phenotype. So it increases in the population, whereas the extremes die out. So when and why does this happen? Well, it happens when an environment isn't changing. When an environment stays stable, it selects for the organisms that are best adapted to that environment, who are surviving and reproducing the best in that case. It's not about a selection pressure here, really. The selection pressure is who can survive the best in this circumstance. Okay, so that's all the things we're going to mention. When is it happening? What happens? Why it happens. Okay, so I've typed out my answer because it's a slightly long one, and I want to make sure that I've got all the points in there. So stabilising selection occurs when the environment is stable and not changing. That means I've talked about the circumstances under which it takes place. The extreme phenotypes are selected against, and these phenotypes die out. This reduces the range, but the mean stays the same. So this is describing what's happening. So the extreme phenotypes are selected against. And then this is my why. So this is my explain part. This is because the average phenotype is best adapted to the environmental conditions, and so that phenotype increases in frequency in the population. So I've got my when, I've got my how, and I've got my why. So I've covered all my bases, and I've said enough for 5 marks for sure. So here we go. We've got the occurs in an unchanging environment. You had to have that because that is the circumstances under which it takes place, which was directly mentioned in the question. The mean is the best adapted, we said that. Selection against extreme phenotypes, or extreme phenotypes die out, we said that. The mean, the median, or the mode does not change or is unaltered. You can say any of those 3 or say all of them, that's fine. The range is reduced, or the standard deviation is reduced. An increasing proportion of populations becomes well adapted to the environment. We didn't really say this. We sort of said that the frequency of the best adapted phenotype becomes increased in the population. We didn't actually say that the whole population becomes better adapted to the environment, but you could say that. But either way, we got our 5 marks without that point. Okay, our second 5 marker then, and our last question of the paper. A mutation can lead to the production of a non-functional enzyme. Explain how. Okay, so we've got another description or a kind of process sort of question here, but it's about explaining. So this is one of those ones where it's like, okay, so this happens, a mutation. How do we get from this mutation to a non-functional enzyme? From one thing to another. well, this happens. Okay, so then this happens, and then this happens, and then this happens, and then this happens, right? So a flow diagram or a process of this would be really helpful. So we need to start with mutation and what is it? And then we need to get to the idea of a non-functional enzyme. What do we mean by that? Well, it's an enzyme that can't form ES complexes or enzyme substrate complexes. So how could a mutation, which is a change in the base sequence of DNA, lead to an enzyme that cannot form enzyme substrate complexes? Start with that, start with the definition of mutation, know that that's where we're trying to get to, and let's think about how we can link these two things together. Enzymes are proteins. Proteins have a structure. That structure is determined by the sequence of amino acids, which is determined by the sequence of bases. We've just said we're changing the base sequence of DNA. So this is really a protein structure question, but you need to go back a little bit further to what controls the structure of a protein, and that is the base sequence of DNA. So change in the base sequence of DNA, otherwise known as mutation, causes a change in the amino acid sequence, which changes the enzyme. Okay, that's not 6 marks. How does it change the enzyme? Okay, it's going to change the sequence or the placement of the R groups, because if the sequence of amino acids is changed, the R groups will be different. If the R groups are different, what does that mean? How does that affect our structure? Well, the R groups are what control the bonds. So the ionic bonds, the disulfide bridges, the hydrogen bonding between the R groups controls the tertiary structure. So this changes the tertiary structure of the enzyme. What do we mean by tertiary structure? Well, obviously we mean the active site. And if we change the active site, then it's no longer complementary shape to the substrate, and so we can't form an enzyme substrate complex. And there we go. We've got to our end point. And this is definitely enough for 6 marks then. So writing out processes like this, where it's like knock-on effects of what happens if something is changed, is really important and is really helpful for these types of questions, because this is what they want to see. They want to see, do you understand what mutation is? Do you understand how an enzyme is coded for by DNA? Do you understand how the 3D structure of a protein is controlled? And then do you understand what happens if we change that? So we've got my long answer is written out in sentences. You could probably get away with writing most of this as bullet points, as long as you're including the key words. So a mutation changes the base sequence of DNA, which we need, which changes the amino acid sequence of the enzyme, which we need. The different sequence of amino acids would mean the R groups are in different places, which we need. This will change the placement of the ionic and disulfide bonds, we need. This changes the tertiary structure of the enzyme, we need. And like I said before, we're going to talk about tertiary structure and we're going to talk about active site, because we need both of them in this case, especially if we're going to make sure we get all 6 marks. Always talk about both if you can. The active site shape is changed, so it's no longer complementary to the substrate, so no ES complexes can form. And the enzyme does not function, which we obviously need to say so that we can link it to the question. Really, if you stopped at like ES complexes not being formed, then that would be fine. But saying the enzyme doesn't function says that you know that you're linking those two things together. Okay, this wasn't hard in terms of putting this together. The language is what's really important, and the steps and sequences and not missing anything out is what's really important. So getting stuff like this down on flashcards and things like that to make sure you understand the processes. Things like this, protein synthesis, big things like natural selection, like we just looked at, the process of natural selection, the process of the different types of selection, inhaling and exhaling of air. All of these are questions that tend to come up as these long answer questions at the end. They're not about application. They're just, do you know your content and do you know your keywords and do you know your kind of processes? Okay, so this is pretty much exactly what I just said. There's not really much room for error here. There's 6 marks, there's 6 points. You need all of this to get the full set. And this is what often these questions are like. There's not much room for error. It will be what you learn, the sequence that you learn, and the content that you learn, and nothing much extra. Change in the base sequence of DNA, change in the amino acid sequence. You can say change in the primary structure, that's fine. Or you can say different amino acids would be coded for, that's also fine. Change in the hydrogen, ionic, or disulfide bonds. You can have any of those, but you have to name one of those type of bonds. Can't just say bonds. Change in the tertiary structure, as I said, changes the active site shape. Those two are separate points, so you need to say both, as I said. The substrate is then not complementary, or it cannot bind, or no ES complexes form. Any of those for that last mark is fine as well. I hope you did well on those last two questions, but if you didn't, then this is definitely something that we should start practicing. Extended response questions are hard, but only if you don't have all those content and that keywords ready to go for stuff like that. Hopefully this was helpful, and definitely hopefully if you've done both papers now, you've got some areas of content that you might be weak on that you need to go and revise, types of questions that you find difficult that you need to go and revise, like the extended response, all the maths, all the graph questions, and you can take this away and now start using this to build your revision plan for the holidays.
Ouch! This is why in some videos I have unexplained scratches.